Welcome to another interesting session of Histology Labs. This is Excellent Grades Academy. This is Dr. Byson EM. Today, we're looking at Epithelial Tissue 1, Histology Practical number 2. But in the lab manual, it is Histology Practical number 3. Without wasting further time, let's get into the session and see what is required of us. So the introduction is that you must know what a tissue is, because today we're looking at tissues. A tissue is a group or layer of cells that work together to perform a specific function. Body tissues are classified into 4 types, and these 4 types of tissues include epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Despite the body's complexity, there are only 4 basic tissues. Today we're looking at epithelial tissue. So now, the epithelial tissue forms continuous sheets of cells that line internal surfaces, cover the external surface of the body, and forms glands. It is a selective barrier that protects tissues and is often involved in absorption and secretion. As we go further in this lab, we're going to see the various surfaces, body surfaces, where epithelial tissue is found, and you're going to appreciate how it lines internal surfaces. So a basement membrane separates an epithelium from the underlying connective tissue. Remember that all epithelial tissue or epithelial cells lie on a basement membrane. So the cells that make up epithelium have 3 principal characteristics. Number 1, they are closely opposed and adhere to one another by means of specific cell-to-cell adhesion molecules that form specialized cell junctions. They exhibit morphological polarity. They have a free surface or an apical domain, and they have a basal domain. They also have lateral surfaces that faces the sides of adjacent cells. The adjacent cells are connected together by tight junctions. Alright, so we need to understand that the basal surface attaches these cells to a basement membrane. Okay, so now the epithelium also forms the secretory portion of glands in their ducts. Now, in this lab, we're not going to talk about epithelium that is found in glands. Rather, that topic is reserved for the next video that we're going to make in lab session number 4. In addition to specialized epithelial cells, such as those that are found in glands, epithelial cells also function as receptors for special senses, such as smell, taste, hearing, and vision. The properties of each domain are determined by specific lipids and integral membrane proteins. Alright, so now this is a schematic diagram that is showing our epithelial cells. So where the cursor is here, these purple cells here are epithelial cells. So these epithelial cells, as you can see, this is cell number 1. It is joined to cell number 2 by means of tight junctions. This is an apical pore, and this is a basal pore. This is a nucleus here. You discover that the basal pore of epithelial cells lie on what we call the basement membrane. The basement membrane has got 2 parts. It has the basal lamina and the reticular lamina. So those are the characteristics of epithelial cells. Now here I want to show you how epithelial cells are opposed or adhere to one another. So you see, this is an epithelial cell and this is an epithelial cell. They are held together by means of cellular junctions or tight junctions. Okay, so if you watch our video on an introduction to histology and epithelial, covering epithelia, you find the names of these junctions. Okay, zona occludens, zona adherens, hemidesmosomes, desmosomes, and tight junctions. Always remember, even when you are drawing epithelia, that epithelia lies on a basement membrane. Okay, so now, what are some of the tissues and their functions? So epithelia that is found in the skin is used for protection. Epithelia that is found in the thyroid is used for secretion. Epithelia that lines the intestines is used for absorption. Epithelia that lines the lungs is used for protection and gaseous exchange. Epithelia in the liver is used for secretion, and the epithelia in the kidney is for filtration. So you can see just from this schematic diagram that epithelia serves various roles in the body. Okay. Now let's get into our lab. The learning objectives for this lab is that after this presentation, you are supposed to, number one, define what a tissue is. Name the 4 basic or fundamental tissues and their functions. List the general characteristics of the epithelial tissue and its functions. Describe the location, the structure, and the functions of the different junction complexes. Number 5, to name the components and functions of the basement membrane. So these are our outcomes after this lab. 6, to give a classification of the epithelia and to describe the criteria on which classification is based. 7, list the location of the different types of epithelia. 8, to name and identify the various cell surface specializations. And lastly, to name some pathological conditions that affect epithelial tissue. Now let's look at the slides that we're going to study in this lab. Okay, so number 1, we're going to do simple squamous epithelia. And the simple squamous epithelia, we're going to look at them where they are found in the aorta and in the vena cava. First of all, you must identify the 2 vessels, both of which are lined by simple squamous epithelia. The simple squamous epithelia that are found in blood vessels are known as endothelium. So your task is to make low power and high power sketches to show a few cells of this epithelia. Now you need to look for a suitable example of the vasa vasorum. The vasa vasorum are blood vessels that take blood to the blood vessels. So they are tiny blood vessels that supplies the muscles of the blood vessels. The blood vessels that supply the blood vessels are what we call vasa vasorum. All right, so you should draw at high power to show the endothelial cells. So draw at high power to show the endothelial cells. So first of all, you should make sketches of the diagrams that I'm going to show you at low power and high power. And secondly, draw at high power to show the endothelial cells, specifically to show the endothelial cells. So now, the following is a diagram that shows the artery, which is the aorta, and the vena cava. So what you will notice is that the blood vessel, the general structure of a blood vessel, is that it's made up of the tunica intima, which is the innermost part, the tunica media, which is the muscle part, and the tunica externa, which is the connective tissue part that protects the blood vessel. Now, the difference between the artery and the vein is that the artery has got a thick tunica media. This is the tunica media here. So this is the tunica media, and the tunica media is a layer that consists of muscles. So your artery has got more muscles than the veins. Arteries are more muscular than veins. As you can see, because it has more muscles, even the lumen here is not collapsed. You can follow it. But when you look at the vein, the tunica media is not as thick as that of the artery. So the vein has got a thin muscle layer, and the lumen here the lumen, the whitish part, is collapsed. So the lumen for the veins are collapsed. Characteristic of veins and arteries that veins are collapsed, arteries are regular in shape. So your task number 1, draw this structure and draw this structure here. Alright, that's task number 1 on the slide. And then task number 2, we want you to draw these walls of the aorta at high power. So here, where this arrow is pointing, number 1, this is what we call the endothelium. The endothelium is simple squamous epithelium. So show the simple squamous epithelium. Number 2 here, this is the internal elastic lamina. Okay, the internal elastic lamina. Number 3 are the elastic fibers. Number 4 are the vasovassorum. So these are just blood vessels. You can just draw them as circular vessels. Number 5 is the tunica intima. So this is the innermost layer. This is the tunica media. And 7 is the tunica externa or the tunica adventitia. So remember, draw the artery and the vein at low power and high power, and then draw one diagram that is showing the endothelium at high power here. All right, so this is the wall of the vena cava. This is the vein. This shows the endothelium, the simple squamous epithelium. Number 2 is the internal elastic lamina. Number 3 is the elastic fibers. Number 4, the tunica intima, tunica media, and tunica adventitia. When you compare the two, the artery has got a larger tunica media than the vein. Let's move on. Let's go to slide 74, which is the kidney. So here you must identify the glomerula scattered in the cortex of the kidney. The task is to draw at high power and low power to show the simple squamous epithelium of the Bowman's capsule. So draw two diagrams, one at low power, the other one at high power. So now, the one that is on the left here, this is low power. And now you may ask, where are the Bowman's capsule? This is the Bowman's capsule here. So this is the Bowman's capsule here. This is the Bowman's capsule here. Okay, so that's what you should focus on drawing. So just show the Bowman's capsule at low power. And at high power, you see this is the high power. This is the Bowman's capsule. So this is simple squamous epithelium. Okay, this is simple squamous epithelium. Draw and label. Okay, so this is a clear diagram here. Okay, this is a clear diagram. Okay, so this is the Bowman's capsule. So these what you see here, this lining here, these are simple squamous, the basement membrane, the epithelia of the Bowman's capsule. This is made up of simple squamous epithelia. So draw this and label this at high power. Okay. Now let's look at the simple cuboidal epithelium that are found in the kidneys. So the task here is to draw examples of the simple cuboidal epithelium in both the transverse section and the longitudinal section views from the renal tubules. So you should draw two diagrams at high power only. So for this one, just draw the diagrams at high power. So this is what we are drawing. The one on the left, this is the transverse view. So draw this. These are cuboidal cells. How do I know that these are cuboidal cells? Because the nucleus here is on the center. It's central. The nucleus is central. So this is high power of the transverse view. So just draw two cells from here. Draw two cells. And then, so draw this cell here and this cell here. The other one on the right, this is a longitudinal section. As you can see, it's wider than it is. It's long than it is high. So these that you see here are cuboidal cells. So this is a nucleus, 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 nucleus. How do you know that this is cuboidal epithelium, simple cuboidal epithelium? Because the nucleus are on the center here. and it's only one layer of nuclear. Nuclear here, 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 and nuclear there. So just draw this one cell and the other cell here. Then you are done. So this is simple cuboidal epithelium that is found in the renal tubules. Okay, now let's move on to slide 55, which is simple columnar epithelia that is found in the ileum. So number one, you should identify finger-like villi on the luminal surface, which are covered by a simple columnar epithelia with a brush border. Now the brush border represents a structure on top of the cells. It is a cell surface specialization called microvilli. So you should observe that the nuclei lie in the basal portion of the cells. Now the task is to draw at low power and to draw at high power a few cells. So you're making two diagrams from this slide that I'm showing you. Okay. So now these are what we call villi. So this is the villi here. Okay. This is the so this is the submucosa rather. Okay. So this whole thing here is the small intestines. So this is a submucosa. Okay. And then this is submucosa, the submucosa that forms hills. This is what we call the Plicae secularis. The Plicae secularis. So now you can see that this is the Plicae secularis here, and there's another Plicae secularis here. And then this Plicae secularis has got finger-like projections. These finger-like projections. The finger-like projections are what we call villi. Okay. Now these villi, on top of the villi, you find epithelial cells that are called simple columnar cells. And on top of those simple columnar cells, you find microvilli. So this image here is low power. So this is low power small intestines, Plicae secularis in the small intestines. So first of all, draw this at low power, and then we go to high power now. So this is a high power view of the small intestines. So remember, these were the villi. So the villi have just been amplified. So this is the villi here, and this villi has got these cells, this cell here, this cell here, this cell here, these cells here. Those cells are what we call simple columnar cells, and these simple columnar cells have got a specialization on top here that appears like the structure of a brush. This brush border is what we call the microvilli. So what I want you to do in this lab is to draw this villi. So you draw this villi here, and you draw this villi here. Okay, so between these villi, there's this structure called the crypt. It forms a gland. We're going to talk about it in the next lab. So after you draw a villi, show these cells on top of the villi. These cells are what we call simple columnar epithelia. So when you, as you can see here, these cells here, these are simple columnar epithelia. When you draw this simple columnar epithelia, of course on a basement membrane, also show the microvilli on top of the simple columnar epithelia. So that's what you are going to draw on high power. Draw a part of the villi, but specifically show the simple columnar epithelial cells. On top of the simple columnar epithelial cells, you should show the microvilli. Okay. So this is the same thing I've been talking about. So draw, show the microvilli, and then draw the simple columnar cells. Remember, this lab is about showing the epithelial cells. So the most important thing is about showing the simple columnar cells with microvilli. Okay. The brush border is just the microvilli that is found on top of the simple columnar cells. Let's move on to slide 75, which are the kidneys. So this section is similar to the slide that we've seen, but it has been stained by periodic acid skiff, which demonstrates polysaccharides, shown here as deep pink lines surrounding each nephric tubule, which is the basement membrane. So your task here is to sketch at low power, okay, or medium power. So just draw one diagram. Okay. So this one diagram here is just showing you this. This is the proximal convoluted tubule. Okay. So these are cuboidal cells and they have a brush border. Okay. A brush border. So just draw one structure here, this structure, and then draw these cuboidal cells and then show the brush border on top of the cuboidal cells, simple cuboidal cells or low columnar cells. They are low columnar cells or they are simple cuboidal cells. Let's move on and let's look at the pseudostratified columnar epithelia. Pseudostratified columnar epithelia is found in the trachea. Okay, so now this pseudostratified columnar epithelia is ciliated. Number one, the one that is ciliated is a type of respiratory epithelia, and it shows nuclei that are piled up in irregular rows with indistinct cell boundaries. So at high power, the cilia are visible on the luminal surfaces. The second one, another example of pseudostratified columnar epithelia, is the one that is found in the epididymis. The epididymis is found in the testes. That's where sperms are stored. So the pseudostratified columnar epithelia in the epididymis has got what we call stereocilia. So for the one that is in the epididymis, just draw them at high power. Just draw them at high power. Okay, so now let's look at this. So this is a pseudostratified columnar epithelia that is found in the respiratory tract. Okay, so as you can see, this epithelia, you can't tell whether the nucleus is in one row. Okay, so it looks like it is stratified epithelium, but it also looks like the cells are on the same level. The nucleus are on different levels. So we don't really know whether it is simple epithelium or it is stratified epithelium. For this reason, it is referred to as pseudo-stratified epithelium. So draw this structure. Just choose 1, 2, 3, 4 cells. Draw these cells like this part here, but include the cilia on top. Include the cilia on top. Make sure when you're drawing this, the nucleus are not at the same level. The nucleus are supposed to be at different levels. Okay, this is the pseudostratified columnar epithelia that is found in the epididymis. So this one here is the epididymis. What you are able to see here, these cells here, these dark things here, this is the nucleus. The dark things are the nucleus. They're pseudostratified, and the nucleus have got these projections here that are called the stereocilia. You can see from this section here, you even have sperm. Okay, this is sperm. Let's shift our focus to here. So this is sperm. Sperm is stored in the epididymis. Okay. And the pseudostratified columnar epithelia here has got stereocilia. So just choose one portion here, draw it, and make sure you include the stereocilia as well. Let's move on to slide number 77, which is transitional epithelia. Transitional epithelia is also known as urothelium. Urothelium is found in the urinary bladder. So here you should draw at low power, and then after drawing at low power, you should draw a portion of it at high power. So 2 diagrams. So you should study it at very much at high power and observe. Pardon me. So observe the umbrella shaped cells. Okay. So it is umbrella shaped. Let's look at this. So as you can see, this is the umbrella shape. This is the umbrella shape. So this is at low power. Draw this at low power, and then at high power it appears like this. So this is what we call transitional epithelium. The reason why it is known as transitional epithelium is because this epithelium can appear stratified as it is now when the bladder is empty, but it will change shape when the bladder is full. So that's why it's called transitional epithelium. It is able to transition. Let's move on to stratified squamous epithelia, which can either be keratinized or non-keratinized. Keratinized stratified squamous epithelia is found on the skin, and non-keratinized is found in the esophagus and in the vagina. So the task is to draw at high power and low power. So in the vagina, you draw at high power and low power, and you look at the ridges that are formed in the vagina and also the muscular layer. So this is it. So this is the esophagus. The esophagus is lined by this epithelia here. This epithelia is what we call the stratified, non-keratinized squamous epithelia. You're supposed to draw it like this. Okay, so it's on the basement membrane, and then you draw it like this. So stratified, non-keratinized epithelium found in the esophagus. So this is at low power, this is at high power. In the vagina, this is at low power. Okay, so this is the vagina. So draw this. So these are ridges that are found in the vagina. This is the lamina propria, and this is the stratified squamous non-keratinized epithelia that is found in the vagina. So make sure you show the stratified squamous non-keratinized epithelia that is found in the vagina. You can either draw that one that is found in the esophagus or that one that is found in the vagina. But for high power drawings, I recommend this one that is found in the vagina, because this is a very good diagram that will show you what you want to draw. Alright, so this is the esophagus at high power. So as you can see, these are the ridges, and then this is the non-keratinized stratified squamous epithelia. It is very, very similar to that one that is found in the vagina. But in the vagina, there were no glands that were shown here. Rather, in the esophagus, you have got glands. So draw one in the vagina and draw one in the esophagus as well. All right. Okay. So this is still the esophagus. So stratified squamous non-keratinized epithelia. This is the lamina propria. This is the muscularis mucosae. All right. And then you've got esophageal glands. These are the esophageal glands. So in the esophagus, you find glands. In the vagina, you don't find any glands. That is the difference. Now let's look at the keratinized stratified squamous epithelia that is found in the skin. So you're required to draw at low power only for this one. So this is the one that is found in the skin. So as you can see, this is the stratified squamous epithelia. So this is the stratified squamous epithelia. This dark thing on top here, that's keratin. So when you're drawing for the stratified keratinized squamous epithelium of the skin, you're supposed to show keratin on top of the squamous cells. So a dark thing is supposed to cover your stratified epithelial cells. It's different from non-keratinized squamous. The non-keratinized doesn't have a dark thing overlying the squamous cells, but for the keratinized, keratin will overlie the squamous cells. Okay, So this is the same thing that we were talking about. Okay, so this is now keratin that will overlie your squamous cells. So draw this and include keratin as well. It's very important. That was the slide, the last slide that we are doing in this lab. So after you do that lab, you label and you draw everything. You're supposed to explain why the transitional epithelium is unique to the urinary system. Why is it only found in the urinary system? And then with well-labeled diagrams, describe the location, structure, and functions of different junction complexes in epithelial tissue. So junction complexes. Between the esophagus and the stomach, there's a junction there. Squamocolumnar junction. Okay, between the anal canal and the rectum, there's a junction there. The anal canal and the rectum, there's a junction there. Between the vagina and the uterus, there's a junction there. So now correlate the function of the following organs and the epithelial linings. Vagina, what is the function of the epithelia there? Ileum, function. Trachea, function. So the ileum is for absorption. Trachea, capillaries, vas deferens, and the alveoli. And then you list 5 different cells in the respiratory system and their functions. And then with 2 examples, differentiate metaplasia versus dysplasia. So this is simply the change of shape in epithelia. Okay, so you're going to see the differences between metaplasia and dysplasia. With a well-labeled diagram, differentiate microvilli from villi. And number 7, with a well-labeled diagram, explain the relationship between cilia and basal bodies related. Okay, so if you saw any abbreviations like PAS in the presentation, that simply stands for periodic acid skiff. For your private study and for further understanding of epithelium, you can watch our video on epithelium. It's already uploaded to this channel, and you can study the chapter in Janquilla, which is chapter 4. This is Dr. Byson EM, always trying to make histology labs and all the topics in medicine very easy for you. Don't forget to subscribe. I'll see you in the next lab.